Thanks for the introduction. And so today I'm going to tell you about our work developing methods to test an approach called RNA interference for manipulating genes in zebra mussels. So this is a collaboration between a number of researchers, including my group at the University of Minnesota Genomics Center, and also Dr. Mike McCartney and Dr. Scott Ballantyne at the University of Wisconsin River Falls. And Lindsay and Margaret are two scientists in my group here in the UMGC that have contributed a lot to this work. So today I'm going to tell you about the approach that we're taking to develop RNA interference tools to manipulate gene expression in zebra mussels. I'm going to tell you about how we're using information from the zebra mussel genome, which was another project that we did a few years back to acquire that sequence information, how we're using that to design these and pick our target genes. And then a lot of the work that we've been doing over the past year or two has been on developing methods for actually testing the effects of these genetic manipulations in zebra mussels in the lab. So I'll tell you some about the kind of our work on developing delivery methods and also phenotypic assays or tests to try to quantify any potential effects. And then at the end of the presentation, I'll tell you a little bit about our initial results that we've been getting this summer on testing these RNAi constructs. So first, the approach. So the idea of doing genetic biocontrol, and actually even the application of genetic biocontrol actually dates back quite a ways. And one example of that, and one particularly successful example of using genetic biocontrol to curb the spread of an undesirable species is this application of the sterile insect technique to control screwworms in the southern United States. And so the way that this approach works is that so screwworms are a kind of devastating agricultural parasite that lay their eggs in small cuts or wounds in livestock animals, or in some cases in humans. And then the larvae actually, crawl around and eat the animal and cause these kind of devastating injuries. And so this was a big problem in the kind of early 20th century in the southern U.S., big agricultural pests. And this approach was developed to basically sterilize male flies, the flies that lay these screwworms, to sterilize the males using radiation, and then release them into the wild, where they would then mate with females, compete and mate with females, and lead to females that didn't produce any offspring. And so this approach was first used sort of in the southern US in the late 1950s. And then the screwworms were gradually pushed south through Central America and to the Isthmus of Panama, where to this day a barrier is maintained. So this process goes on every week, where in a factory down in Panama, flies are sterilized, dropped from an airplane. 15 million sterile male flies are dropped from an airplane along the Isthmus of Panama to maintain this barrier to prevent the screwworms from spreading north. And so more recently, developments of genetic technologies like CRISPR have made the process of potentially producing these genetic biocontrol organisms much simpler. And so there are a number of approaches that have been developed, including this engineered genetic incompatibility approach that was developed by Mike Smansky's lab here at the University of Minnesota. And then also gene drive approaches, which are being tested and applied in a variety of organisms, most prominently in mosquitoes. And so these approaches, so gene drives use CRISPR to bias the inheritance of a trait. So you basically cause a trait to rapidly spread through a population. And one approach that's being used in mosquitoes is to take a trait like sex determination and bias the population to produce, say, only males. And that will eventually, lead to a crash in the population. And so, these techniques, I think, hold a lot of promise for genetic biocontrol of a variety of species. And but unfortunately, because of some of the limitations of working with zebra mussels, we can't apply these types of heritable genetic techniques to zebra mussels because we really aren't able to grow them over the long term and generation over generation in the lab. There is a project that's going on, funded by MASERC right now, and led by Ben Minerick from Minnesota Zoo. So Ben is a scientist and aquaculturist who has a lot of experience growing mussels and other aquatic organisms in the lab. And so he's been working on trying to solve some of these problems in propagating zebra mussels. And there's actually there's a video on the MASERC website that I've got the link to here. And if you're going to be joining us for the lab tours tomorrow, in addition to walking through our lab and seeing where we're doing these RNAi experiments, I'll also walk you through Ben's lab and show you some of the things that he's doing to try to solve this problem and allow us to culture mussels. But right now, at least, we're faced with the question of how can we genetically manipulate an organism that we can't robustly cultivate? So we need a method that doesn't rely on making a heritable change to actually the zebra mussel's DNA itself, because even if we made such a change, we couldn't grow it to the next generation. So instead, we've turned to an approach called RNA-interference. And this is a method that was first discovered in this nematode worm called C. elegans. And it was shown that double-stranded RNA induced a gene silencing response in this organism. So an example of that is shown here on the bottom. So this is in this panel that's labeled panel E. You can see an expression of a protein called GFP, or green fluorescent protein, in the worm. And on the right-hand side, this protein the expression of this gene has been knocked down using double-stranded RNA against this GFP gene. And one of the really interesting things about this technique, and one of the intriguing things for our purposes of trying to deliver this to zebra mussels, is that this double-stranded RNA interference works both when you inject it into the organism, but also when you feed these worms bacteria that express these double-stranded RNAs. So this already suggests a potential delivery mechanism where we could, induce expression of double-stranded RNA in bacteria, put it into the water, and then have the zebra mussels filter it out and hopefully take up that double-stranded RNA expressing bacteria to induce this effect. So RNAi turns out to be a very generalized mechanism that exists across essentially all eukaryotic organisms, and it's been used in a variety of species to induce various mutant phenotypes. So, for instance, this is an example in fruit flies or planaria, but also importantly in other bivalves like oysters. So this is this oyster example, they actually were able to decrease the volume of the gonad with an injected RNAi construct. And in fact, this mechanism actually also exists in humans, and there are now even FDA-approved therapies that are based on RNA interference. This process is thought to be sort of an ancient antiviral defense mechanism. So double-stranded RNAs typically don't exist within the cell, but there are viruses that have double-stranded RNA genomes. And so this double-stranded RNA material, when it enters the cell, is recognized as foreign. There are a number of enzymes that chop it up and then look for anything that's similar to that double-stranded RNA and degrade it. And so essentially what we're trying to do here is use this RNAi machinery to trick the zebra mussels into mounting this gene silencing response against their own critical genes. And so we know from our work sequencing the zebra mussel genome that the kind of core machinery for carrying out this RNAi process is present in zebra mussels. So all these different enzymes that chop up the RNA and degrade anything that is similar, these exist in the zebra mussel genome. So the approach that we're taking right now is we're taking zebra mussel target genes, cloning them into a expression plasmid that can produce double-stranded RNA, putting this into bacteria and causing the bacteria to make a large quantity of that double-stranded RNA, and then feeding these bacteria to zebra mussels, and then looking for effects, looking for either changes in their growth or behaviors, or also we can also look at the knockdown of these genes. So we can look molecularly and see, did this double-stranded RNA actually cause the expression of that gene in zebra mussels to go down? So the advantages of this approach, for our purposes, first of all, this gives us a means to manipulate the biology of the animal at all, given the limitations in kind of growing and maintaining them in the lab. But it also has the potential to be really highly species specific. So the RNAi effects rely on the complementary base pairing between these RNA molecules, and those sequences can be very specific to an organism. And this is in contrast to chemical controls, where these are often very broad spectrum and have a lot of undesirable off-target effects. It also potentially has really flexible options for deployment. So assuming that this approach works, one could potentially deploy this either as a highly specific chemical agent. So that would be, using the double-stranded RNA itself as the control. Or one could deliver it in inactivated microbes, or even in replicating microbes, which might have the advantage of kind of persisting in the environment for a longer period of time. Or there's also the possibility that one could pre-treat zebra mussels, sort of analogous to that sterile insect technique, where you pre-treat them to give them something that will make them sterile or will sort of poison the natural population. And you could release those pre-treated mussels into the lake. And the advantage of that is that you would actually not be releasing anything that's genetically that has any sort of heritable genetic modification. So it would be a potentially very safe way of delivering this in a lake. So next I'm going to tell you about the target genes that we're currently looking at, and how we've gone about choosing them, using information from the zebra mussel genome. So the Zebra Mussel Genome Project was initiated by Dr. Mike McCartney, and was a very large collaborative effort between a number of different groups here at the University of Minnesota, including scientists from MASERC, from the University of Minnesota Genomics Center, and from the Supercomputing Institute. And then we also had a number of external collaborators as well who helped us with different aspects of this project. And so we made use of a variety of the really cutting edge DNA sequencing technologies that we have here in the University of Minnesota Genomics Center to assemble a really high quality reference genome. We assembled this, we generated this chromosome scale assembly where we ended up with 16 sort of scaffolds, or which correspond to the 16 chromosomes that zebra mussels were known to have from other sort of karyotyping methods. And then once we had that genome sequence, we needed to annotate the genome and start to try to understand where the genes are, what they are, and how they function. And so we were able to use take a few different approaches to doing that annotation. So the first one is sort of using information that we know about the genetic code, which is this code that translates DNA into proteins, and that's conserved really across the tree of life. So we know from this genetic code that there are certain start codons, there are certain triplets that indicate the start of a coding sequence. And then we also know that there are a number of stop codons. So we can look for what are called open reading frames, which are basically long stretches of these triplets that could potentially code for a protein. And so these open reading frames would then be sort of hypothetical genes or hypothetical coding sequences. We can then use information. So we have lots of other genome sequences that are available some of which have been really well characterized. For instance, the human genome, where there's been more than 20 years of effort on trying to understand the function of all the different genes in the human genome, but also model organisms like fruit flies or mice. And so we can start to ask, for each of these open reading frames, do they have, are they similar to anything that we've seen before in other genomes? So this one that I've highlighted here, if you align this to the human genome, you actually see that it has a lot of similarity to the serotonin receptor. And I highlight this one because this is actually an interesting target gene for us. So zebra mussels, so serotonin is, of course, an important neurotransmitter. Many of you are probably aware it's involved in mood disorders in humans, like depression. And it's actually a signal that zebra mussels use to control spawning, or the release of their sperm and eggs into the water. So we can actually take mussels from the lake, bring them into the lab, put them in jars, and squirt in a little bit of serotonin. And within 45 minutes or an hour, we'll start to see them releasing sperm and eggs. And so this is an attractive target for potential manipulation, because if we could knock down the expression of this receptor, one could potentially prevent them from receiving this cue to release their sperm and eggs and possibly disrupt spawning. And then the other way that we can look at the genes in the genome and try to get some evidence about, whether they're interesting targets or what their functions might be, is we can look directly at gene expression. So we basically can take different tissues, grind them up, isolate the RNA, and then sequence it, and we'll actually see what genes are being turned on in those tissues. And so in the context of the Genome Project, we did this for a few different tissues, including the mantle, which is what excretes the shell of the zebra mussel. And so this is a list here of the top 50 or so, or top I don't know, 30 or so most highly expressed genes in the mantle. So these are potential targets. If we wanted to go after shell formation, these would be potential targets that we could go after. So we have a lot of different information from the genome and from kind of other comparisons that have allowed us to generate a large list of potential candidate genes that are involved in processes like shell formation, or attachment to surfaces, or reproduction, or just basic cell biology and kind of health, that we can begin to target as potential genes that could be manipulated to induce an effect, or induce some sort of fitness defect in zebra mussels. So we've kind of prioritized this larger list that we have and begun to actually clone these constructs. And this is work that was done by Lindsay and Margaret in my group, and then also Reardon Smith at the University of Wisconsin River Falls. And we've now made about 30 or so of these double-stranded RNA expressing constructs and put them into bacteria, so we can begin to test the effects of these genes in zebra mussels. And these targets that we've initially zeroed in on, some of them are just involved in general kind of cell maintenance and survival. So things like involved in very basic biological processes, like the ability to make proteins or degrade them, or neurotransmission, or things that are really essential for the health of an organism. And then we've also cloned a large number of the genes that are involved in forming the byssal thread attachment fibers that zebra mussels use to attach to surfaces. And then also we've been going after the serotonin signaling pathway as a way to potentially manipulate that spawning process. And then, so in order to test these constructs, we need phenotypic assays, or ways of sort of measuring the effects that these constructs have. So one of them that we've been doing a lot of work on, we kind of jokingly call zebra mussel Yahtzee. And it's really a simple test where we detach a mussel from its substrate, put them in a jar, let them sit for a certain period of time, and then we tip the jar over and see how many mussels have attached to the surface. And we've shown that the mussels will kind of repetitively do this over many days. So if we detach them each morning, come back later, either later in the day or the next morning, they'll continue to reattach to the surfaces. And so, so we want to see, if this assay is actually reflecting some aspect of muscle health. So we did an experiment where we treated the muscles with different concentrations of copper sulfate. So this is one of those chemical controls that are sometimes used to try to control zebra mussels. And we showed that with increasing concentrations of copper sulfate, the percentage of zebra mussels that reattach to the surface decrease. So we think that in addition to being able to use this as a means to kind of test some of those attachment fiber genes that we're looking at, if we impair the health of the mussel, if we kill the mussel, they're also not going to be able to reattach to the surface. And so we think this assay can be used to kind of screen a broad swath of these constructs and test for generally for defects in muscle health. We're also looking at their ability to filter things out of the media. So we have this other test where we add bacteria to the media, and these might be the RNAi constructs that we're dosing the mussels with. And we take a sample of the water right after we've added the bacteria, and then we plate that out on a plate. We make tenfold dilutions and then plate those out as spots on a plate. And you can see these spots appear at these different dilution levels. And then we wait a period of time and take another sample of the water, say 12 hours later. And you can see that when there are no mussels present, those spots look about the same as they did when we initially put the dose in. But with the mussels present, they've taken some of that bacteria and filtered it out of the water. And so the amount of bacteria decreases. So this is a way that we can measure both the amount of these RNA bacteria that they're taking up and their ability to sort of filter things out of the water. So we're actually working on a number of different tests in addition to this attachment and filtering test. We're also hoping to be able to measure growth over time. We found that this is really dependent on the diet. So we don't really see much growth using sort of an inactivated shellfish diet that's typically used to maintain them. But when we include live algae, we start to see more growth of the mussels. So this will be an important thing to optimize in the future, and it's part of what Ben Minerick has been working on. And then we're also hoping to develop assays for spawning. So this is where we take mussels and add serotonin and get them to release their gametes. This assay might have some seasonality to it. So we found, and it's been reported in the literature that that there's a peak of spawning early in the summer, and we've also found that we saw a lot of spawning induced by serotonin early in the summer, but as the summer went on, we see less and less. And then Mike McCartney has also been working on these histological assays that allow us to section the mussels and quantify the amount of gonad that's present. So we could use this if we're knocking down some of these genes that are required for reproduction to quantify effects on the development of the gonad. So just in the last few minutes here, I'll tell you a little bit about some of the initial tests that we've done this summer. So these tests have been done using this reattachment assay where we put the mussels in jars, let them attach, and then add in the dose of RNAi bacteria and then test the effect on attachment. What we're hoping to see is something like this graph on the right, which is sort of schematic data, where we see a certain level of attachment in the control, and we might see certain genes that are similar to the control, but then we find genes that cause strong defects in attachment. So this is the actual design of the experiments that we've been conducting. And so what we do is we play zebra mussel Yahtzee and measure this baseline attachment prior to adding a dose, so we get a sense of how well those mussels are attaching. And then we do 3 to 5 days of replacing the water in the jar while adding in these bacteria that express the double-stranded RNA. And then each day we're also measuring the attachment as well in these mussels that have been dosed. And then we do a washout period where we just add water without the bacteria, and again, measure attachment post-dosing. And so this is kind of what the data looks like. And I should have Lindsay's picture on this slide as well. Lindsay Engelbach is, she's been doing a lot of these experiments throughout the summer. And you can see from the time course here, these are sort of heroic experiments where she's coming in every day, including on the weekends, and measuring the attachment of these zebra mussels. And so what you can see is that when we have a no bacteria control, or a control where we're adding in double-stranded RNA against a target of GFP, this green fluorescent protein, which is a jellyfish protein that's not present in zebra mussels, we don't see any effects on attachment. You know, they kind of bounce around a little bit, but they hover around 80 or 90%. And so then we can begin to test different targets. So this blue line here is showing one of our targets. This is a gene that's involved in proteasome function or protein degradation, which is a really basic and essential process in most cells. And so, you know, what we can see is that, you know, we see this maybe this little blip post dosing period. But again, these numbers kind of bounce around a little bit. And so what we really need to do is, you know, kind of repeat these assays multiple times and try to see, you know, if these kinds of effects are robust, or if there's sort of a one-off fluctuation in the data. So far we've tested a handful of these constructs. And this is sort of just taking in the average attachment around that dosing period. So the green bars here are the no bacteria controls, or the GFP controls. And then these other color bars are different constructs that we've tested, which include this ubiquitin gene, some of the foot proteins or the attachment fiber proteins, and then also a translation factor called CPB4. And this star data point is the one that I showed you in that previous graph, where you can see, you know, we saw a little bit of a decrease in attachment, but when we repeated that experiment a second time, we saw it looked pretty similar to the controls. And so, you know, we know there's a little bit of noise and variability in these measurements. And so, so far, you know, among the handful of genes that we've tested, we're not seeing these very large effects on attachment, at least. So what we're hoping to do in the future is, you know, we've got about 30 of these constructs. We're hoping to, this summer at least, to get through screening as many of them as we can in the attachment assay, to see if, you know, we can find a few of them that do have large effects. We also want to, you know, do some molecular work to verify that we're not actually knocking down the expression of the gene. And then in the future, we're hoping to start to use some of these other assays, filtering, growth, reproduction, to also test the RNAi constructs and see if we can see effects in other systems. Ultimately, the system that we're using to deliver these RNAi constructs right now is not something that we could apply in a field setting. So they require induction with the chemical, the bacteria require induction with a chemical inducer to produce these double-stranded RNAs. So we also want to start working on, just thinking about the future, working on delivery vectors that have, that produce these double-stranded RNAs continuously, so, as a way of building something that could potentially be deployed in the field. And actually, one of the angles that we're exploring there is whether we can use this algae called Chlamydomonas reinhardtii, which is a non-pathogenic algae that exists in a lot of natural soils and water. It's got this grass status, or generally regarded as safe, which means you can basically eat it. So it's a safe thing to use. There's a lot of expertise here in the University of Minnesota campus in working with this algae. So there's actually what's called the Chlamydomonas Resource Center, which has lots of different strains of algae. And importantly, they can be genetically modified to produce double-stranded RNA. So we think, you know, in the long term, using something like this, which is a more natural food source for zebra mussels, and something that, you know, we can still use to produce these double-stranded RNAs, might be a better way of delivering it. So how could this eventually aid in management of zebra mussels? Well, the first is indirectly. So, if we can get this RNAi approach working in the lab, it gives us a means to start manipulating zebra mussels and kind of understanding more about their biology and really kind of studying the effects of these different genes. But the nice thing about the approach is that in addition to developing a tool that's useful in the lab, it also takes a step toward developing something that could potentially be deployed in the field as well, using one of these different approaches that I had mentioned earlier, either as a chemical agent or as sort of delivered through a microbe. So with that, I'd just like to thank all my colleagues in the University of Minnesota Genomics Center and all of our collaborators on this project, especially Mike McCartney and Scott Ballantyne, who have both done a lot of work in helping me to lead this project. And I'm happy to take any questions.
Thank you so much, Daryl. We do have a few questions and I can read them for you to make it easier. April asks, why did you use 2 controls in your zebra mussel Yahtzee for attachment?
Yeah, so, we view those controls as being slightly different from one another. So in the one case, we're not adding any bacteria at all. So we just have the mussels kind of sitting in water, and we're changing out the water similar to what we do with the dosing. But you could imagine that the bacteria themselves might have an effect. And as we've been working on these assays, we've actually found that can be the case. So we've tested a wide range of doses to try to see and use that filtration assay to see where we see the best kind of uptake of bacteria by the zebra mussels, or at least the best depletion of the bacteria out of the water. And what we found is that as we kind of increase the dose and keep increasing it, if we get to we can get to a certain point where there's so much bacteria there that it starts to kind of coat the walls of the jar and starts to cause attachment defects on its own. So that's where that GFP RNAi control is useful, because it's basically mimicking adding the bacteria, and we're able to control for whether there's an effect of just having the bacteria there itself, independent of the double-stranded RNA expression.
Thanks for that. We've got another one here. So it says, this anonymous attendee says, so less attachment equals reduction in reproduction, or less attachment equals fewer problems that zebras cause by attachment to things?
So it could be a few things. So so you could affect attachment in a few different ways. So one is that you could directly impact their ability to produce those attachment fibers and actually attach to a surface. That could have impacts on their dispersal, their ability to attach to the boat and move to the other lake, say. But it's also known that they seem to require attachment to really be healthy. So they won't filter very well or kind of grow very well if they're not able to attach to a surface. So you can directly impair that attachment process. But in those zebra mussel Yahtzee experiments, attachment is a proxy both for their ability to make those attachment fibers, but also potentially for the health of the mussel itself. So we can use it to kind of cast a wide net and screen for all kinds of defects. But ultimately, some of those defects may be really specific to attachment, and some of them might be having broader impacts on the health of the mussel.
Thanks for that. Why is it difficult to rear and maintain zebra mussels in the lab?
Yeah, I mean, it's kind of a ironic thing, given how well they survive and thrive out in the lakes and rivers. We've actually joked that we should just go put up signs saying like "experiment in progress" by all the lakes, and maybe that would be enough to wipe them out. But so there are a few different a few different blocks. So we're able to go out and collect mussels and actually keep them in the lab for a fairly long period of time, for sure several months. And I don't actually know what the upper limit is for kind of maintaining adult mussels in the lab. But the big problems come in trying to get them to go through their reproductive cycle. So we know, we can get them to spawn using that serotonin trick, and we can get sperm and eggs, and potentially even fertilize them to make embryos. But then there's also there are challenges in sort of growing them from that very early stage, and having the larva survive. And so that's one of the problems that Ben is working on is how do you properly grow up those larvae? And he actually has some devices in his lab now that are similar to the tanks that are used to cultivate like jellyfish. And they have this like upwelling effect that allows the larva to kind of stay suspended in the water like they would in the lake. So there's part of those like tricks of like how do you kind of like mimic the conditions. And then there's also just sort of like generally keeping them healthy and figuring out what the right diet is. So that's another thing Ben is working on, is trying to figure out, what algae or what mix of algae do we need to be feeding them. He actually has a little like kind of mini indoor pond set up that tries to kind of mimic the environment of a lake with some plants and other things in it that is used to kind of cycle through water into the tanks to try to make sort of like a more natural environment for the mussels. But yeah, it's mostly kind of getting all the way through that reproductive cycle and producing the next generation of mussels from adults that you have is really the biggest bottleneck.
And what are the steps involved in taking technologies like this from the lab to the field? And maybe what is the regulatory process like?
That's a good question. And for a lot of these new genetic techniques, the regulatory processes are still being defined. That's certainly the case with these CRISPR technologies and gene drive. There is actually a little bit of a precedent for an RNAi approach. So there has recently been a successful application of RNAi to control an agricultural pest that targets corn. And so so there has been like EPA approval to use this engineered corn that employs an RNAi mechanism to defend against a pest. I think that will actually be an important future part of this project. So, right now we're really focused on a proof of concept for the technology, right? So does this work, and is it kind of viable as something that you could actually use? But then I think there's going to be a lot of work that needs to be done on both defining the regulatory pathway and engaging with stakeholders and the public, and making sure that this can be done in a way that is safe, effective, and takes into consideration the concerns of all the stakeholders.
Thank you. We have just one more question. Could approaches like this be used to increase populations of a threatened or endangered species, or can it only be enabled to reduce population?
Yeah, that's an interesting question. I think for a lot of the endangered species, it's a lot about habitat. So maybe that might be the most direct way to do it, is like, if we remove zebra mussels, walleyes are going to do better, right? And so you might be able to think about ways of applying it in that sort of way. Certainly the CRISPR technologies and these ability to do genome editing, there's been a lot of discussion about whether those types of approaches could be used to actually even de-extinct species. And there are actually several efforts underway right now where groups are for instance, trying to modify elephant DNA to reintroduce the mammoth or something like that. Those I think those projects all have their technical challenges, but these tools are sufficiently powerful that one can begin to imagine doing things like that.
Thank you so much. If we have any other questions, you can go ahead and enter them in the Q&A button at the bottom. You can wait for those, but in the meantime, I'd like to thank Daryl for the information shared here today.